Hello, my name is Carter White, and in this video I will be explaining our body's most abundant extracellular matrix protein, collagen. To start off, I need to first explain collagen's shape and molecular structure. As we know, this is very important and indicative of how the protein functions. The first thing I'd like to explain is collagen's unique 3D structure. Collagen at its core is made up of 3 alpha chains that form a triple helix of repeating amino acid triplets. I provided a couple pictures of the triple helix here on the left so it's easier to visualize. I have also provided a picture of DNA's double helix to the right, as it might help compare collagen's shape to another important and well-known structure like DNA. There are many differences in this comparison, of course, but the shapes and part of their function might be analyzed better using a familiar molecule like DNA for comparison. Zooming in further to the triple helix structure of collagen, we can see a couple more important features of the molecule. The most important of these features is the repeating peptide triplets that consist of glycine, X, Y, where X and Y represent varying amino acids. More often than not, proline and hydroxyproline are the X and Y respectively, as shown in this image. This picture shows all 3 of these amino acids that might line up in one of the alpha chains within the triple helix. This time reading glycine XY from bottom to top, X being proline and Y being hydroxyproline. If we analyze even further into the specific amino acids, we can see some clear acidic and basic regions of each molecule. Each amino acid has an acidic region at the top where we can see carbon double bonded to an oxygen as well as an OH group. As we know from class, this can provide stability and possible resonance structures if the amino acid is to act as an acid and get rid of the H on the OH. We can also observe a potentially basic amine group at the bottom of glycine and within proline and hydroxyproline as well. These acidic and basic regions make the molecule perfect for forming all sorts of interactions and other with each other and possibly surrounding molecules as well for binding to each other in things like hydrogen bonding interactions. The last part of the structure I'd like to touch on is how it all comes together in a much broader sense. This picture does an excellent job of showing the scales of collagen and summarizes all the structures I've talked about so far. Starting from the smallest structure of amino acid triplets at the top, then it goes into the triple helix, and it's called spirals in this picture. Then into the group of triple helices forming microfibrils, then subfibrils until eventually forming the big macrostructure of a collagen fiber. This property of bundles will help a lot in determining important properties I will discuss later. Now that we know the structure of collagen and how it forms its interactions and structures within structures, we can easily see and predict some of its most defining properties. Some of its properties include cell recognition, controllable mechanical properties within tissues, for example, loosening or tightening a tissue, and biodegradability. However, its most important and unique feature is the ability to form 3D scaffolds of various physical conformations. This ability of collagen allows it to be one of the strongest and toughest structures in the body, and is the primary reason the body loves to use it as a ground substance in many tissues. Going back to the DNA comparison I made earlier, we know that DNA has to have a very stable and strong structure in order to encode our entire genome and hold tightly to itself so that we don't lose or interfere with any of that important code. And it does this through a double helix. Collagen, on the other hand, adds one more strand to that helix, forming a triple helix, which makes it an even stronger molecule. An analogy that could be useful is to think of the triple helix, the fibrils and microfibrils, as hair or threads that make a rope. One hair or string can be easily broken when you pull from both sides of the string. But if you grab many hairs or weave multiple strings into a rope, it becomes very difficult to stretch and break. The more strings or hairs, the tougher it becomes to pull apart. The third string in the triple helix, as well as the microfibrils and fibrils formed by collagen molecules joining together, make the entire molecule extremely difficult to break, which is useful for holding together many moving parts of many living tissues in animals. The image here provided is used as a reference to where collagen is found in these tissues and ground substances, and how it's used to anchor and hold pieces into place. As you can see in this example, many types of collagen exist, shown in dark red or purple in the lamina reticular layer, and blue in the lamina densa and lamina lucida layers. Each collagen is being used to hold the layer of tissue together and to keep it from tearing, disassociating, or moving too far from this original shape. The final topic I want to cover is the synthesis of collagen within the human body. I provided an image to help walk through the process, outlining it has 6 important steps here. The first is not listed, in my mind at least, and it's the transcription and translation of the genes relating to collagen, which come from that nucleus. Then it shows it being delivered to the rough endoplasmic reticulum through mRNA, and then the images steps finally start in this rough ER with the alpha chain backbone of the previously mentioned glycine XY. Then the second step is the hydroxylation or addition of an OH group to the proline, and in this particular example it uses lysine as that Y group rather than the hydroxyproline I mentioned earlier. This step, importantly, requires vitamin C. And then after this step, a sugar is added to lysine, which helps the alpha chains and the triple helix stick together. Then the molecule at this stage is called procollagen, leaves the cell in a process called exocytosis. Once outside of the cell, procollagen is cleaved by an enzyme, and it clips the carbon and nitrogen ends off the molecule. And finally, this collagen is ready to form its cross-links through covalent bonds of hydroxylysine, in this case. In closing, we can use the biological molecule of collagen to see how organic chemistry and life itself are linked, as well as explore another of the infinite cases where structure and properties of a substance directly influence its function. Collagen is the most abundant extracellular protein found in mammalian species, and it's for good reason. It's synthesized by an efficient 6-step process, and it forms structures such as alpha chain triplets that can form into triple helices that can then gather into microfibrils and fibrils and provide many important functions, just like stability, strength, and structure to various tissues all over the human body. Of course, I did not learn all of this information via my own experiments and knowledge, so here are the sources I used to research and formulate this presentation. Thank you for watching.